So yes, I'm going to give you an overview of the models for Duchenne muscular dystrophy. And you've already heard the introduction. So today I was going to give you a very short introduction about Duchenne, but I'll mostly skip that because you've already heard in Micah's presentation and others that the key points. Mostly I want to use Duchenne as an example to illustrate the key points in the choice of animal models. And yes, the thing is I will focus on in vivo and, evaluation. So compounds can obviously be screened in cells, but I will focus on the preclinical evaluation and all the animal models and all the interest of the animal models to understand the biodistribution and efficacy of your compound. So I'll give some example of the outcome measures that can be measured in mouse models after exon skipping treatment. And then I'll mention some other large animal models, and then what's necessary and what's just good to have. So what is really required when you want to go to the clinic. So very, very briefly, Duchenne muscular dystrophy is caused by mutation in the dystrophin gene, so the DMD gene that's been presented before. It is composed by 79 exons, and it encodes various dystrophin isoforms. The one that we will focus on today is the long one, which is called DP427. This is the muscular isoform that's also represented here. And so the exon skipping concept has already been introduced to you. So just very quickly, most of the patients carry deletions of one or several exons. And this is the example of a patient that would carry a deletion of exon 50. And that would induce a disruption of the reading frame and the appearance of a premature stop codon and truncated protein. The exon skipping approach aims at excluding an additional exon, in this case, for example, exon 51, because 49 and 52 are in frame, and this can allow the translation to continue and the synthesis of a slightly shorter protein, but functional or at least quasi-functional, like the one that are found in Becker muscular dystrophy. So this exon skipping approach can be induced using antisense oligonucleotides. And so what are the key points in the choice of an animal model when you want to test this type of therapy? First, there are some points that are common to, let's say, all the therapeutic strategies, not only splice switching or oligonucleotides. Generally, when you want to test a therapeutic strategy on it for a genetic disease, you want your animal model to have the similar genetic basis as the patient. That means the same gene has to be invalidated. So for Duchenne muscular dystrophy, the model needs to have no dystrophin expression. You also want the phenotype to resemble the human pathology. So for DMD, this includes muscle weakness, respiratory insufficiency, a cardiomyopathy. And the disease progression should be well characterized, and ideally you would like to have already reference values available to allow you precise outcome analysis. But then you also have some specifics to the RNA therapeutics fields, and for example, the splice switching approach. In our case, you need the type of mutation to be very similar to the mutation that are found in patients. So for example, you cannot have a mouse model that's a null model, so you wouldn't have the expression of the mRNA because you want to act on the RNA. So if it's a null model, like it's completely a knockout, that's inappropriate for your type of therapy. And for DMD, you ideally want similar exon deletion that you would find in patient, although you'll see that some of the common model are not exactly this. The first step is generally to do a proof of principle in mouse model, and we'll see that they are very easy and they are the most commonly used animal model because they are easy to breed and generally commercially available. So I'll go through the classical MDX model, but also some more severe model, some larger model like the rat, and even larger model like dog and pigs. And finally, the need to have human specific model, so potentially transgenic model with humanized gene. So the MDX is the most commonly used mouse model for DMD. Maike already presented it quickly. So it was found and described in 84. The main phenotype is relatively mild. So this is the main difference with patients. And as Maike said before, it only has a slightly reduced lifespan. So it's almost normal. And it stays ambulant, so it can walk its entire life. It does have some respiratory pathology and abnormal cardiac function, but only at older age. What does make it a good model, and especially for exon skipping, is that the mutation is suitable for exon skipping approach. So it's a mutation in the exon 23. It's a nonsense mutation, so it induces stop codon in exon 23. And luckily, exon 23 is in frame. So if you start targeting splice sites of the exon 23, you can skip it and link 22 and 24, and that would result in a quasi-dystrophin that's lacking only exon 23, which is only a small coding sequence. So what can you do in this type of mouse model? What you want to achieve using an animal model, and especially a mouse model, is to be able to evaluate pharmacokinetic of your compound in vivo, assuming that you have already proven that in cells it's working. You want to see its biodistribution, and you want to see the pharmacodynamics, so the efficacy. So biodistribution, generally following the intended administration method. So for Duchenne, it is generally a systemic, either intravenous or subcutaneous. And then you want to be able to look at the molecular evaluation at the RNA level, at the protein level, eventually some biomarkers that would have been described. And if the phenotype is there, you can do a functional evaluation. And we've seen before, and I'll show you again what you can do in this type of mice model, looking at the muscle force, cardiac, respiratory function. And you can also have an idea of a preliminary evaluation of the safety profile that gives you an idea of the tolerability of your compound in mouse model. But here you have to be careful and take into account the disease phenotype. And I'll show you with dystrophic mice, for example, they have some biomarkers that are elevated, so they may not be ideal for this. This is just a quick slide to show you that some examples that I will give today are based on this particular chemistry of antisense oligonucleotide, which are called tricyclo-DNA. And so, for example, when we use these tricyclo-DNA antisense oligonucleotides in the MDX mice, the in vivo injection allowed us to check for the tolerability, but also the biodistribution. So in the initial experiments, we did intravenous injection or subcutaneous injection, and you can quantify the oligonucleotide by LCMS, for example, and have an idea of how the compound behave in the bloodstream. And after the end of your treatment, you can have an idea of the biodistribution in the different tissues. So for us, we're more interested in the muscles. So we collected all the different skeletal muscle, but also diaphragm, heart. And it was very interesting for us to see that even in the brain, we could detect the oligonucleotide, which was very interesting. So this gives you an idea of the pharmacokinetics and the biodistribution in a disease model. In Duchenne, it is particularly important because the biodistribution is generally facilitated because of the disease. So that would give you a better idea than treating a wild type mice. And this may be true for a lot of disease model. You want to get an idea of the efficacy, and in this case, we had compared our particular chemistry to two other chemistries, and you can see that we had a look at the exon skipping level, and we had a look at the protein level, and in each case, we were here able to show that the tricyclo were giving a higher exon skipping levels and higher dystrophin restoration. So this is important to be able to show the efficacy in your disease model. And then when the phenotype is there, you can start to look at functional outcome. So for a disease like Duchenne muscular dystrophy, you would like to show muscle function. In MDX, we said that the phenotype is relatively mild, but as Maike showed, you can still do quite a lot of functional tests. One of them is the treadmill, so you can force the mice to run, and they generally do not run as long as the wild type mice, as you can see here. So that allows you to evaluate your treatment to see if you can rescue that. There's a lot of tests like the grid tests, where you place the mice on an inverted grid test and you can see how long the mice can stay. And generally, the dystrophic mice do not stay as long as the wild type. You can use the rotor rod, again, for muscle function, even though it can, as Maike said before, this is also dependent on the behavior of the mice. So when you start looking into functional assessment, you have to be really careful about potential other defects that the mice may have regarding the behavior. And this is a specific aspect that Maike mentioned in a short scientific mission where she did electrophysiology on the diaphragm. Here, for example, this is electrophysiology on the tibialis interior. And it's slightly different because in this case, the muscle is still attached to the mouse. So it's not an isolated muscle, but both type of measures are very informative. And you can see that in this case, as in the case that Maike presented, the specific force is much decreased in the diseased animal. You can have a look at the respiratory function using what we call plethysmography, where you can place the mice here in cages that measures all the pressures, and that gives you a lot of different measures, such as the inspiratory time, expiratory time, and the respiratory function is impaired in MDX mice. You can see the curve here are quite different from the healthy mice. And I've given you here the example of the minute volume, for example. And you can see that the minute volume is significantly reduced in MDX compared to wild type. And this is a way of showing that your treatment had an effect on the respiratory muscle. You can also look at the cardiac function. But as I said, this is doable, but this is also a limitation of the MDX mice because you need to work only in older mice. So when the mice are younger or less than 6 months, it is very difficult to detect this impairment. And you can see actually these mice here were about 6 months, and you can see the dispersion, the variability between the animal. And it's already at that age quite difficult to show a real defect, even though with a large group of animal you can. But this is where the limitation of the severity and the mildness actually of the phenotype is a limitation for the treatment. So there are a lot of other mutants that have been created, and this is the example of one of them, especially for cardiac function. So the CMA dystrophin deficient MDX that have been used especially to look at treatment in the heart. So for example, here in the group of Matthew Wood, when they were looking at peptide PMO treatments to improve the cardiac phenotype. So in order to aggravate the pathology of the MDX model, there has been a lot of efforts. So some of them, for example, have tried to backcross the MDX on different genetic background. And for example, the DBA mice has the cross with this background has clearly worsened the dystrophic phenotype. So this particular mouse model is still an MDX, just on a different background, has fewer myofibers, increased fibrosis and fat accumulation. Unfortunately, it's not a good model for cardiomyopathy. But a lot of labs are currently using this mice because it is more severe. There has also been a lot of double mutants created. So the mice, the MDX is only mutated in the dystrophin gene, but people have crossed them with mice that were invalidated for MyoD or dystrobrevin or integrin. And you can see here, for example, This is from a nice review on the animal models where it is schematized the lifespan of human and patient, which is fortunately a little bit higher now. And you can see here the different model where the MDX is not really reduced lifespan compared to all of these different models. One of the double mutants that's been quite widely used is called the double knockout, and it's actually a knockout, well, an invalidation of the utrophin gene, and you can see it's here. No, where is it? It's here. It's approximately 3 months, and we've used them also with tricyclo. You can see what is nice with a mouse that has a severe phenotype is that it's really visual. So you can definitely see the phenotype. The mice has a lifespan of about 10 to 12 weeks, and it looks like that already from 8 weeks of age. So you can clearly see the effect of your treatment when you start treating the mice, and it is something that you can see straight away. The other advantage is that you can do other type of measurements, just like looking at the active time, because here it would be very obvious that the double knockout are clearly much impaired compared to the wild type or the distance traveled. And you can do all the other tests that we are also doing in the MDX. One of the limitations of a double knockout model or a model that is too severe is that you need to start the treatment extremely early. And here in that case, we had to start treatment from 4 weeks old. And that's doesn't allow a great window of therapeutic action. So, for example, in human, you would have weeks and months to treat the patient, and here you don't have that time. So you need somehow you need a treatment that's a little bit stronger. So there are other limitations of the double mutants because obviously they look like they are a better phenocopy of the dystrophic phenotype. The fact that this is a genetic, a double mutation, then the genetic is different from what you have in patients. Obviously, DMD patients do not have, they do not lack both dystrophin and utrophin or dystrophin or myogene. So genetically speaking, they are not a perfect model. But arguably, the nonsense mutation in the exon 23 of the common DMD mouse is also not ideal and the best representative of patients. So this is why other models have been created with the deletions of exon that would be genetically closer to DMD. That's the case of a transgenic mice called the MDX52. So this mice has a deletion. It's a knock-in, so it doesn't have the exon 52. And it's very useful for the skipping of exon 51 or 53. So it's a DMD patient-like mutation that was engineered quite a while ago, but it's starting to get more interest. And the pathology is quite similar to the MDX. So I would say that the only advantage is that you would at least target exon 51 or 53 when you're doing exon skipping. But you still have to be careful because it's still the mouse sequence. And so that's a point that we'll see at the end. If you want to use human-specific sequence. Other rodent models that have been generated to try to get more severe phenotype are the rat models. So one of the first rat was created with a deletion in exon 23 by a French group in 2014. And it does look like the rats are more, indeed more severe. So they have muscular strength and spontaneous activity decreased. They clearly have necrosis and regeneration associated with progressive replacement of fibrotic and adipose tissue. So they look like they mimic the DMD phenotype a bit more. More recently, and I don't think the paper is published yet, but there's another rat model that's been created with a deletion of exon 52. So again, very suitable for exon 51 and 53. And it also is a good model for cardiomyopathy. So apparently the rat presents a more severe cardiomyopathy phenotype. Interestingly, a bit more recently, in 2018, there's a rabbit model created using the CRISPR-Cas9. And again, it is also a severe phenotype. Here, it's not a problem, but it can be a limitation. There's some of the rabbits, some of the neonates died in the first 2 weeks of birth, and approximately half of them still die quite young. So, and this is still not very well understood why they would die so young, although it's a good model for cardiomyopathy. But in that case, you may wonder, and that's one of the topic that we should discuss is what's the advantage of having a rabbit over a rat, for example. Like the phenotype looks quite similar. Some people may argue that rabbits are easy to breed. So then it also depends on your animal facility and what's easier for you to have as a model. Finally, there are larger models. The dog model, there has a number of different breeds of dog that have been characterized and reported with dystrophin deficiency. One of the most common and one of the most famous is the GRMD dog model. So it's a golden retriever dog. Also, it's been backcrossed on I think that's a beagle. That is the CXMD in Japan. So the dogs are actually really severe. But again, there's a lot of dog-to-dog variation, and some of the dogs die with the first 6 months of life, and some others behave well, survive and live much better. Interestingly, it's a splice site mutation, and that leads to the natural skipping of exon 7. So exon 7 is not included in the messenger RNA. So in terms of exon skipping, this impose, you can see here, to restore the reading frame, the skipping of 2 exons. So if you want to restore the reading frame, you absolutely need to skip both exon 6 and 8. So this has pros and cons, like all the animal models. It is nice if you want to be able to show that double exon skipping is possible. But if you're, for example, if your compound and if your clinical trial is only going to be about 1 compound, then it is a challenge to use the dog in that case. There are other dog models that have been reported, in particular the Cavalier-King Charles panel with a splice mutation that results in exon 50 deletion. So here it is useful because it is amenable to exon 51 skipping. It has a severe phenotype as well, and given the small size of the breed, it may actually be a more valuable model for the exon skipping approach. And finally, and this will be the last large model I mentioned, and it's good because it's already been described this morning. So the pig model was generated a number of years now, in 1992, in Munich. And so the dog has, no, I didn't change this, sorry. So the muscular strength and spontaneous activity are clearly decreased in the pig. Sorry, there's been an inversion here. And so I'm not sure actually if there has been many trial of exon skipping. I know that we've done proof of principle both with tricyclo and the 2' orbital. So there are people in the action that are involved with the pig. And I remember that we're trying to back cross them on mini pigs. So it would be interesting to know if they managed to get that. And finally, one of the important thing is when you want to move forward with your compound, it's a specific human compound, you may want to have transgenic models that are carrying the human dystrophin gene. And in the case of DMD, there's a very useful model called the HDMD mice that was created at LUMC. And it carries the entire human DMD gene. And so this, in the end, you have two models, one co-expressing the murine and human dystrophin, and the HDMD that has been crossed with the MDX mice, in which case he only express the human DP427, but you have to remember that it would also express the other isoforms of the mouse dystrophin. So yes, as I mentioned, it's a very useful model to evaluate the exon skipping level. In that case, this is a wild-type phenotype because the human dystrophin compensate for the MDX pathology. So you cannot look at protein rescue. And when you are skipping, you're skipping an exon from a wild-type mRNA. You may be subjected to NMD, and also the fact that you may have decreased uptake in wild-type muscle. But since then, the LUMC group has created a humanized, so from the HDMD, deleted the exon 52. So again, applicable for exon 51 and 53 skipping. So this type of model is very effective and very useful. So to summarize some of the key points, in Duchenne, we have a lot of animal models that are available. And it's very important to know exactly what you're looking for in your model. And all these models are available to allow you to validate the efficacy and tolerability of your compound. So for example, a specific chemistry or a new ligand. And generally you would use the mouse surrogate for this. If the phenotype is appropriate, you can demonstrate the functional improvements. And finally, it's very useful to have humanized model to validate human-specific sequences. But really, you can wonder what is really required to move to the clinic. So is it really required to test your molecule in the dog model or in the pig model? From a regulatory point of view, you will need full regulatory tox studies, and generally for oligonucleotides, 2 species are required, 1 species of rodent and generally non-human primates. There's no model of Duchenne for human primates, so you would have to go through the tox studies. It's important to remember, so when you are at this stage, this is extremely expensive. So generally you partner up with a company or you get a lot of funding to do these studies. It is still possible to do preliminary tox studies in non-human primates with only a couple of animals. That would cost less, and that would help you to know the efficacy of your compound. So the key messages and the take-home messages are that the genetic basis of the disease should be the same in your animal model than in patients. The model should reiterate the key hallmark of the human disease, and the phenotype has to be quite robust and reproducible over generation. One absolutely crucial thing is that the animals should be readily available, ideally commercially available, easy to handle and maintainable. So this is why mouse models are much more used, or rat, than dog models or pig models. And as I mentioned at the beginning, the disease progression should be very well characterized so you can have reference values available. One final point is that you really need to know what you want to use the model for. Is it to show the efficacy of your compound? Is it to assess functional outcome? Is it to assess the sequence specificity, in which case the humanized model are useful? Is it a preliminary stage where you want to screen antisense sequences? In that case, you might be interested in reporter mouse model. And I just wanted to show you, this is the last slide about a reporter mouse model that exists for Duchenne, where the DMD gene has been fused to GFP, and then the mouse has been back-crossed with the MDX. So that allows you to look at the dystrophin revertant fibers, for example, just by looking at the GFP. And when you're doing a treatment, you can actually look at the mice treated in vivo. So you can do live imaging and look at the dystrophin live after treatment, like here, for example. So this can be a very useful mouse model when you want to look longitudinally So you do not need to sacrifice the animal, and you can do live imaging during the course of the treatment. So I'll finish here. I'm sorry, I think I lasted a bit longer than expected. So feel free to come and ask me a question later if we don't have time now. So thank you all for your attention.